Hello students, today let us study about smut disease in corn. In my last videos, we had studied about covered smut and loose smut in case of wheat. Today let us study about the different kinds of smut diseases we can come across in corn. Two different kinds of smut diseases can be observed in corn. One is corn smut, which is caused by Eustylago medis, head smut, which is caused by Spasilotheca riliana. These are the two different diseases which are going to study in detail in this particular video. Coming to the symptoms of corn smut, as we all know, this is one of the diseases caused by Eustylago medis. This is one such organism which belongs to subdivision Basidiomycotina, class Teleomycetes, order Uridinales, family Uridinaceae, and the species genus which we are studying is Eustylago, and the species which is going to cause corn smut or a common smut in case of corn is called as medis, Eustylago medis. This particular pathogen is airborne. It's an airborne, and the primary infection takes place onto the aerial part of the corn plant. So it causes localized infection. This we have to remember. Once the teleospores land on the surface of the host, that is on the aerial part, or any aerial part, either on the leaf or on the leaf sheath or on the stem or on the tassel or so-called inflorescence or a head or on the corn, if it lands, they cause infection only to that particular part or particular tissue. That's called as localized infection. As I was teaching you before, systemic infection, where you can come across in those loose smuts as well as in covered smut, where actually the infection takes place to the entire part of the plant, that is called as systemic infection. Whereas corn smut, that is infection caused by Eustylago medis, is one of a infection. It's called as localized infection. That is, once the symptoms goes on like this, symptoms are most commonly noticed when the ear produce tumors or galls. It is the formation of the tumors, as you can see in the images which I have put on the right side of this particular slide. You can see the formation of tumors or galls. What are these tumors? That is mass of cells. They are called as undifferentiated mass of cells, that is called as tumors. Such kind of tumor formation happen wherever the infection has occurred. The infection can happen even onto the entire inflorescence, or it can happen on the leaf or the leaf sheath or on the stalk, etc. Second symptom is, here once the infection has happened, there will be a formation of swelling. That is, wherever the tumor has been formed, that particular cell undergoes the process of hypertrophy or hyperplasia. Hypertrophy or hyperplasia. These two terminologies you have to remember. What is this hypertrophy or hyperplasia? That is, it is the process of the increase in the size of the cell or the number of the cell. That particular process is called as hypertrophy or hyperplasia. So here the same process are happening, where once the infection happen, those cells, once the pathogen enters into the cell, there will be certain hormonal imbalance, that is the secretion of hormones in that particular host cells alters, which leads to the hypertrophy and hyperplasia. That is, increase in the size of the cell or the number of the cells. Those processes are called as hypertrophy and hyperplasia respectively. Here later, so wherever the infection has happened, there the the swelling happen, which start beginning as small whitish grayish irregularities which expands and turn black as they fill with the spores. Once the infection has happened, there will be a change in the mass or the volume or the size of the cells in the host, which begins to grow. At the same time there will be a fillings inside that particular cell with black spores. Those those irregular mass of cells, they are called as galls. Each of the galls can grow up to 4 to 5 inches in diameter, and in the same region there will be an accumulation of n number of spores, that is teleospores. You can see in this particular images the size of the normal corn grain when compared to that of a infected corn. Infected corn size will be comparatively more than that of a normal, and if you see the color, they are blackish in color. That is due to the production of spores inside them. Here, once the spores get accumulated in each of such corn seeds, the the outermost wall ruptures and releases large amount of teleospores or smut spores into the surrounding. These fungal spores are blown by wind for considerable distance, and it can infect new plants. Galls, as I was telling, galls can be formed even on the leaves or in the stem, all those, and even they can remain. These galls, once they ruptures, they can remain for some time, and eventually they become hard and they will dry once all the spores have been released. And another important thing which we have to note down over here is this corn or the smuts which has been infected to the corn and causing the galls. They are commercially important. They are commonly called as corn truffles or Mexican truffles, which they use like as the other mushrooms, because of the presence of rich protein in them, and also because the presence of an amino acid called as lysine, which normally the corn lacks. So thus, because of this property, this particular galls, the infected corns, have been found to be extensively used as corn truffles, and in Mexico it has been traded as Huitlacoche. It has been traded as Huitlacoche, where it has got a highest commercial value because of its medicinal properties, as well as because of the rich content in the protein, and also because of the particular odor it has got, because of which it has been extensively used. Remember, this is being important for the point of examination. Edible mushroom Huitlacoche, which has been traded as Huitlacoche in Mexico. These are nothing but the fungus which has infected the corn, which is the fungus Eustylago medis. I have also given the link about this Huitlacoche. You can go through that. So once again I will repeat, the symptoms of this particular disease are like this. Symptoms, which as I was mentioning, one of the airborne disease where the localized infection happen to any part of the plant, which leads to the formation of galls, where the size of the cells or the mass of the cells increases, that is hypertrophy or hyperplasia happen, which leads to the increase in the size of the cells, and inside these cells the fungus grows and produces teleospores, and these cells which has been infected, they grow to 4 to 5 inches in diameter, and once the large amount of spores has been occupied inside, the membrane ruptures and the spores are released. These smut spores, once they are released or blown out, they get disseminated through wind once it come in contact with the healthy plant, and the cycle can continues. The immature galls, that is before the formation of this smut spores, before the process of dehiscence, before the process of rupturing, this galls are also edible, which are called as corn truffles, which is been has got high medicinal values and also has got a rich protein in them. So that's the reason it has been traded extensively and has got high value, economical importance, and also with respect to economics also has got good value. Then coming to the life cycle of this Eustylago medis, the primary infection takes place by the spores called as teleospores. Teleospores are thick walled, they are blackish or brownish or sometimes olive green in color. They are made up of thick wall layer. You can see the spores, dikaryotic spores, dikaryotic spores called as teleospores in the image of this image A. You just concentrate on A. These are the teleospores, and as I was mentioning, they are round in shape, made up of two wall layer, exine and intine. Exine is ornamented and it is dikaryotic in nature, made up of two nucleus. This teleospore has got high resistance power. It can withstand high temperature, and also it's in dormant phase where even due to the lack of nutrition also they can survive in the soil, and it can be there in the soil, and once it's overwinters during the summer season or during the spring season, what happen during favorable condition, this teleospore, the nucleus of this teleospore undergoes the process of karyogamy, to form a diploid cell. This diploid cell, during favorable condition, it start germinating. How it's germinate? It absorbs some amount of moisture. Because of the absorption of moisture, turgor pressure is created inside, because of which exine rupture, intine produces germ tube. At the same time, the diploid nucleus undergoes the process of meiosis. Soon after the process of meiosis, the nucleus, all the four nucleus of opposite strains, they enter into this germ tube. The germ tube can be called as epibasidia. These epibasidia undergoes the process of septation. From each cell of this epibasidium pinches out basidiospores. Some mycologists also called as them as sporidium. It's also called as sporidium. The sporidium, once they have been formed, these are the one which causes primary infection to that of a corn plant. Once this basidiospores come and land on the surface of the host, the haploid basidiospore, they come in contact with the another basidiospore or the sporidium of opposite strain. They undergo a process of fusion, or sometime each of the basidiospore, once they land on the surface of the host, they produces germ tube. Basidiospore, I am talking about the basidiospore of opposite strain. Here we have represented positive and negative strains which has been landed on the surface of the host. In the figure D, you can see the basidiospores of opposite strains have landed. They produces germ tube. They can directly enter into the host tissue by the penetration of the cell wall, or through the injuries or through the stomata. They can enter into the host cell. The mycelium of opposite strain undergoes the process of somatogamy. Remember, the primary hyphae, once the basidiospore germinate, they produces primary hyphae. they are haploid. Primary hyphae of opposite strain undergoes the process of somatogamy. What is the meaning of somatogamy? The fusion of the cytoplasm, whereas the fusion of the nucleus is delayed, which leads to the formation of dikaryotic cell. So such cell germinate and produces secondary mycelium. The secondary mycelium, they are dikaryotic in nature. This dikaryotic mycelium grow grow inside the host tissue and utilize in between, or they can enter even inside the cell of the host tissue. It can be either intercellular or intracellular. And grain, as we know, the grain is an important economically important part, where it contains large amount of nutrients inside them, and there inside the host cell they grow and and germinate. And they have to, once the nutrients is over, they start producing, or they get transformed into the spores called as teleospores. So you can see at the same time, once they enter inside the host cell, due to the hormonal secretion inside the host cell because of the pathogen, what happens, the cell undergoes the process of division. The rapid division of the cells happen, that is hyperplasia or even hypertrophy can also happen, which leads to the formation of irregular mass of cells. Those are called as galls. Once the nutrition is vanished inside those galls, what happens, all the mycelium which are being formed, they get transformed into the teleospores. These teleospores, which are either brownish or olive green in color, and it is being covered over by a thin delicate membrane. Those structures are called as galls. Once the membrane ruptures, mass of spores are released outside. Those are called as teleospores, which are dikaryotic in nature. This dikaryotic teleospores overwinters, they go for dormancy. Once onset of favorable condition, they can undergo process of karyogamy followed by meiosis, again the formation of epibasidium and the formation of basidiospores. These basidiospores, once they land on the proper host, they produce germ tube, and the germ tube or the primary hyphae of opposite strains undergoes a process of somatogamy, which leads to the formation of secondary mycelium, which are dikaryotic mycelium. This dikaryotic mycelium grow and later they become transformed into the teleospore. At the same time, the host cell or the host tissue produces galls. Inside these galls you can find the teleospores. This is about the disease cycle of Eustachiago. So this is about the common smut or corn smut disease. Now let us study about head smut of corn. This is a disease which is caused by Spasilotheca reliana. Spasilotheca reliana. And this is one of a soil-borne or a seed-borne disease. And here the infection is systemic infection. That is, the infection takes place to the entire part of the plant. Whereas the corn smut, it was localized infection. Wherever the infection happened, only that to that part of the plant, you can see the symptom only over there. Whereas here the infection can happen to the entire part of the plant. This is one of the soil-borne or seed-borne disease. That is, the infecting pathogen or a propagule can be found in the soil or in the seed. So here, like how it was in case of loose smut or in covered smut, till the formation of a head or an inflorescence, no visible symptoms can be found. These infected plants looks like that of healthy plants. Only after the process of the formation of head, there will be no difference between the healthy as well as the infected plant. So head smut, that is this disease, is not evident until the tassel or ear appears. Tassel or ear, nothing but the inflorescence. Here, the head is the infected head I'm talking about. Head is, in this image also you can observe, head is characterized by the presence of sori. That sori in the sense teleospores. It is characterized by the presence of sori on the tassel, ears, or rarely on the leaves. See here, this infection is confined, the symptom is confined only to the inflorescence or onto the tassel. Here, the spores gets accumulated only inside such we can see such kind of black spores or teleospores covered over by a thin grayish membrane, and this grayish membrane soon ruptures to release powdery mass of spores which are quickly scattered by air currents or rain. So once again I will repeat, this is one of the soil-borne or the seed-borne disease. That is, the infected pathogen is found in the soil or on the seed. The infection takes place to the germinating seedling. Once the seedling is germinating, this pathogen also infect the seedling, and it grow along inside the seedling and reaches the inflorescence, where we were studying about loose smut and the cover smut. In the same way, it also infect. Once the inflorescence has been formed, only the symptom can be found in that particular head or on the tassel. So what kind of symptom we can find? The entire tassel. Tassel is the part where actually the corn has to be produced. Here, inside this thallus, each of the grain or the grain or the tassels is being completely occupied by the teleospore, and they have been covered over by a grayish white membrane, and this membrane ruptures and releases powdery mass of spores, which once they are released, they can be scattered by air and rain. And another important symptom is these tangled mass of thread-like strands, which are the vestigials of the vascular system of the corn inflorescence, ramify the sori and are characteristic of infection by the fungus. Infected tassels are completely or partially covered by sorus and normally do not produce pollens. So once the tassel has been infected, so here how it is different from that of a corn smut. So here you can find the entire corn, that is the entire tassel would have got infected. Along with this spores, tangled mass of spores, the thread-like strands. These thread-like strands are nothing but the vascular system of that particular inflorescence, and this infected inflorescence are completely covered by that of teleospores, and such the flowers of that particular maize plant may never produce pollen, and the individual florets are replaced by the teleospores without the formation of galls. That is the characteristic feature of common smut. That is, here the entire the florets, the seed formation doesn't happen at all. Before the formation of the seed, that ovary of that particular flower would have been occupied by that of a teleospores, and that teleospores are released outside. The gall formation is being not found in this symptom. That's the difference between the corn smut and the head smut. Again, coming to the disease cycle. So as I was mentioning, this is one of a seed-borne or the soil-borne disease. These teleospores are the main source of infection. Teleospores are the main source of infection. These teleospores can be present in the soil or in the debris from that of a previously infected crop. if it is found in the soil. These teleospores have the capability of withstanding very high temperature and any of the unfavorable condition it can withstand and stay in the soil for a long time, or from the seed, if the seed has been infected with that of a spores or teleospores, if such seed has been sown, the cycle can continue. So here, as I was mentioning, the teleospores are present in the soil. They overwinter in soil once the seed has been sown. During that particular period, these teleospore, they break the dormancy, they germinate. The nucleus inside the stereospore, as I was mentioning before, in the same way, the nucleus undergoes karyogamy followed by meiosis. Formation of epibasidium happen, septa formation happen, followed by the formation of basidiospores, haploid basidiospores. Haploid basidiospores of opposite strain, they undergoes a process of somatogamy to form a diploid cell. This diploid cell can infect the germinating seedling, and through root system, and later the mycelium develops systematically. That is, the mycelium can grow into the entire part of the plant, but the mycelium concentrates more on the meristematic tissue, that is onto the apical meristem, because that is the region where actually the inflorescence production happen. Once the inflorescence are produced, these mycelium also enters inside this growing inflorescence, enters into the gynoecium part of the inflorescence and utilizes all the nutrition in that particular ovary. Later they get transformed into teleospores, diploid teleospores. These teleospores, and the ovary has been covered over, they have been covered over by a thin membrane. These membrane ruptures and the spores are released outside and the cycle continue. So this is one of a disease which causes the organism causes systemic infection, that you have to remember. It's a soil borne disease or the seed borne disease, infects the root system of the plant and enters inside the host tissue. They grow and develop inside the host tissue, enters into the meristematic region, where the inflorescence or the head formation happen. They enters into the tassel region, infects the ovary, and once the nutrition is over, they get transformed into the teleospores, and they are released outside and the cycle can continue. Coming to the control measures of head smut: use of disease resistant varieties, crop rotation, application of fungicides onto the soil before sowing. As I was mentioning, it is one of the soil borne disease. That is the reason you can treat the soil with appropriate fungicide to destroy the pathogen. Application of protective fungicide, and we can go for seed treatment. As we know, this is also one of a seed borne disease. Seed treatment can be done before the process of sowing the seed. You can apply captain, which is one of a fungicide. However, we can go for carboxin, which is also called as Vitavax, which is one of the systemic fungicide. What is this systemic fungicide? These are some group of fungicide which can enter inside the host tissue and they can destroy the pathogen. Those are called as systemic fungicide. So this carboxin is one of the systemic fungicide. So you can go for carboxin or Vitavax, which is comparatively better than that of a other fungicide. Maintaining proper fertility of the soil, remove the infected part of the plant, and it can be burnt into ashes. That is, maintaining proper sanitary measures in the field. That is, you can remove the infected plants especially the inflorescence or the tassels or the ears which have been infected before the spores are scattered. Before that point of time, you can remove such entire plant can be removed and it can be burnt into ashes. And these are some of the references. Thank you once again for your patience to listen this video lecture. Thank you.